Good morning, everyone. Today we will finish our walk through cellular respiration. We'll be looking at the electron transport coupled with proton pumping across the membrane. And then we're going to see what that proton gradient actually does for the cell, the force of that gradient driving synthesis of ATP. I have one other animation I would recommend anyone with SmartWork take a look at. We'll take a peek at it today. We'll actually look at a computer model of the action of ATP synthase. And we will finish our study of cell respiration. That will be part of our exam on Monday. We will be covering 4 chapters. Chapter 9, evolution of genomes. 11 and 12, the structure and function of membranes. Chapter 3, we looked at energy and then cell respiration, some covered in chapter 13, some in 14. Exam starts at 9:30 on Monday. I got a couple of, I see someone seems to have their mic on. If you can just double check that your mics are off. A couple of pointers about our exam, reminders of what you should expect for the exam. On Monday, I sent out an announcement. Anyone who has any SAS extended time, you should take a peek and make sure that it looks like you have the correct time set up for you. We will have some REEF questions today, so make sure that you have signed into your REEF account. Make sure you have joined our course. And this is our reminder of any of our online exams. It is already published. It's set up in the quizzes tab. You'll see an exam labeled exam 2. It begins at 9:30. It is a timed exam. There's 50, everyone has 50 minutes. What that means is while I have a window set up from 9:30 till 10:25, I've got a 5-minute buffer in there. So that means if you start the exam a couple of minutes late, you hopefully still have your full 50 minutes to take the exam, right? But if you start at 9:30, your timer will count down and you will exit the exam at 10:20. One attempt, you'll see one question at a time. You can't go back. So make sure that you have a timer set up next to you or watch the timer that starts as the quiz, once you begin the quiz, and make sure you're giving yourself enough time for each of those questions. 34 questions in total in your 50 minutes. Right. Remember, I told people that you should be assembling, you should be creating your own review sheets. You can have those physical notes in front of you. You are not allowed to go to other parts of the web course. You're not allowed to go to any other websites. You're not allowed to interact with any other students or anywhere or anybody else. This is an exam. I'm asking you to show me what it is that you know. So you could have a couple review sheets. And again, as I've been telling people, the preparation of a small 1-page review sheet is a really great study tool, right? As you assemble that, you're actually trying to link all of the different concepts that we've been talking about in each of the chapters. That's a great way of preparing for an exam. But don't try to find the time to look through PowerPoints or look through answers. You will not have enough time to be searching through all of your notes for answers. All right, and I will be in the web courses chat room during an exam in case someone has a quick question that you want answered. Usually there aren't any, but I will be there monitoring the situation. Okay. Any questions about that? All right. So where we left off, we were looking at in introducing the concept of the electrons moving through the electron transport chain. And we want to spend just a minute here looking at one of those respiratory complexes, meaning a complex of proteins in that inner membrane, a large multi-enzyme complex, each of them are, and this is one of them. This is the cytochrome oxidase complex, is the last complex in the electron transport chain. And this is actually where oxygen is held, waiting for electrons to be passed to the oxygen to accept those electrons. And just to give you a view of the complexity of this theory, these are two different subunits of a 13 subunit complex, 3 of which are coded for in the mitochondrial genome. So these 2 proteins have been synthesized in the mitochondria and then assemble with other proteins that are transported into the mitochondria that have been synthesized out in the cytoplasm to form just one of these complexes. And what I also want to point out here is these complexes are associated with metal ions. And right here, where these 2 copper atoms reside, is where oxygen, an O2 molecule, is held, waiting for electrons to be passed to it from the upstream complex. And I can even enhance this a little bit, showing you that the actual electron acceptor is an iron atom that can become oxidized and reduced. It accepts the electrons. That iron atom is held in this heme molecule within one of the subunits. And what it does is the oxygen is sitting there waiting until 4 electrons are passed to the iron atoms from the upstream a component of the electron transport chain, and then those electrons are passed to, they're not showing us the oxygens here, are passed to the oxygen. So they're waiting for the electrons. Oxygen, the final electron acceptor that will accept those electrons plus protons and be reduced to water. So let's take a look at more diagrammatically the different components of the electron transport chain. 3 complexes of multi-enzyme complexes with metal atoms, which are the actual electron acceptors, are in the path of the electrons that are passed from NADH. NADH will donate its electrons to the first complex, which then passes to complex 3, which passes to the last complex, cytochrome oxidase. That's the one that we just looked at the ribbon model of. We're going to look at this on the next slide, but we also remember that some electrons have been carried in a different electron carrier, FADH2. And FADH2 passes its electrons first to another complex that we call the iron sulfur complex. And then those electrons are passed to cytochrome BC complex, and then finally to cytochrome oxidase. So in the figure on the next slide, it does not illustrate complex 2, but I'll show you where that lies in this chain of electron acceptors. So let's take a look at, and this is what we had in the short little video on Wednesday. Sitting in the mitochondrial membrane, the inner mitochondrial membrane, we have our 3 complexes. There's the first one, NADH dehydrogenase complex. This is the complex of proteins that will accept the electrons that have been donated by NADH. Right? And our little pink line is showing the path of electrons from NADH, where they will be accepted by O2 at the very end of this electron transport chain, where that oxygen will combine with electrons plus protons to form water. Right? And what we can also see is those electrons pass from NADH to the first complex, then pass to this hydrophobic mobile electron carrier that we call ubiquinone, that will carry it from that carries electrons from complex 1 to complex 3, and then to another mobile electron carrier, cytochrome C, passing to our final complex, the cytochrome oxidase complex. Complex 1, 3, and 4, electrons passed from FADH2 will be passed to a complex that would sit right about here, complex 2, which will then pass the electrons through the rest of the chain as well, finally residing in H2O, in water. And remember, as those electrons are being passed, the acceptance, the reduction, and then oxidation of the electron acceptors within these complexes result in the movement of protons across the membrane from the matrix side, pumped over to the other compartment, the intermembrane space. And so what the mitochondria does is will create a gradient, right? High concentration of protons on this side, low concentration on the matrix side. And that is a potential energy. That is a force. The potential energy of that gradient will allow the flow, diffusion of protons from one side of the membrane to the other, as long as the protons have a place to pass through. And the channel that they can pass through is through a complex that we call ATP synthase, another complex of proteins. The only place across the membrane that the protons can diffuse across. As the protons diffuse across, that will drive movements of this ATP synthase complex, causing a rotary motor to be activated. And that rotary motor will power the synthesis of ATP. And so ATP that is generated in this manner, ADP that is phosphorylated in this manner, we call being generated by oxidative phosphorylation. The redox reactions of the electron transport coupled to the proton pumping drives the synthesis of ATP. So let's take a peek at our ATP synthase on our next slide. Here is our inner mitochondrial membrane, right? Our lipid bilayer. This entire complex here is our ATP synthase. The different components of the ATP synthase form, number one, a channel by which protons can diffuse from the side of high concentration, diffuse to this compartment of lower hydrogen ion concentration, which is the matrix side. And this is actually a very steep gradient. Not only do we have a gradient of difference in concentration on both sides of the membrane, but this membrane, of course, has a voltage across it. So we have an electrical gradient as well. And so along with the chemical gradient and the fact that this membrane is negatively charged on, or has more negative charges on the matrix side than it does on the intermembrane space side, this is a very steep gradient, which means it's a very strong force, right? The potential energy of this movement, the potential for these protons to move is great. And that movement results in the synthesis of ATP. Protons, one at a time, bind to this central portion that rotates. The rotation of this portion will interact with other components of this stator. This is the knob portion down here that will actually drive changes in the polypeptides that result in the synthesis of ATP. This is a rotary motor. It actually is the smallest molecular rotary motor. And what we have is this electrical energy of this proton gradient, excuse me, the chemical energy of this proton gradient is converted to mechanical energy of the rotation of this motor that will drive the synthesis of ATP. So this is where I would like to show us a little piece of a really nice animation of this rotary motor, this molecular turbine in our cells. So give me a second to close this down for a second. Open up our video. And I just have to change the audio for a second.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized. Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft.
Okay, let me get this back. So I think that's very nice to be able to have a view of the actual motion, right? That two-dimensional, just looking at an image of this static image doesn't really does not show what's going on. So I think it's a lot easier to understand what's going on with this ATPase by seeing that computerized modeling of it. Oops, sorry, I don't have my audio right yet. Again. Okay, I should be back now. All right, so let's see if we can answer a question. And somebody has their mic on. If you want to turn it off, please. Everybody check your mic. Ryan, you have your mic on. Thank you. So here's a question. What we've been looking at is the coupling of electron transport with pumping of protons and ATP synthesis. So we're asking here, what if cells are exposed to some sort of drug that allows that inner mitochondrial membrane to be permeable to those protons. What do you think would happen in that case? And make sure that you would agree that the other options here, that those would not occur. How are we doing? Everybody had a chance to vote? All right, let me close it up. And yes, A would be the correct answer. If protons were allowed to freely cross the membrane, what would happen is that electron transport and proton pumping would be uncoupled from ATP synthesis. So ATP synthesis would decrease, but glycolysis would still occur. Citric acid cycle could still run. Carbon dioxide would still be released. Oxygen would still be accepting electrons as they pass the electron transport chain. NADH would still pass its electrons. But really what would be happening is that the fuel would be oxidized, that chemical energy would be converted to heat, right? Heat radiated, released, but very little ATP would actually be generated. Right? So that an uncoupler would reduce ATP synthesis, but still allow oxidation of fuel molecules. And as a matter of fact, infants, as well as hibernating mammals, have uncoupler components in the brown fat, which allows for fuel to be oxidized, generate heat, but not be used to generate ATP. So it's a way of being able to maintain a body temperature during hibernation and in early development by temporarily uncoupling ATP synthesis in some cell types from the oxidation of fuels. So what did we accomplish, or what has the cell accomplished in the process of cellular respiration? And if we are considering in the first case, aerobic respiration, if we're looking at the yield, we want to make sure that we're following per molecule of glucose. So we went through this a little bit earlier, but let's see, in addition to coupling of electron transport to ATP synthesis, what are we making overall? So the first line is how much ATP has been generated both from glycolysis and from the citric acid cycle. Anybody wanted to remind us how much ATP is made during glycolysis per molecule glucose? What's the yield, the net yield of glycolysis? 2 from glycolysis. 2. Thank you. How much ATP is made in the citric acid cycle? 2 more, right? So we have a total of 4 ATP from glycolysis and citric acid cycle. Then in aerobic respiration, in the mitochondria, in the presence of oxygen, more ATP is generated from, as we just saw, oxidative phosphorylation, right? So that's requiring electrons. So where are the electrons coming from? How many molecules of NADH with electrons that it can contribute? How many NADHs are generated during glycolysis? That's a pretty common number, right? We're still on 2, right? Glycolysis generates a net of 2 ATPs and 2 NADHs. Where else do we get NADH from? We get it from oxidation of pyruvate, right? The pyruvate that was a product of glycolysis will be oxidized. How many NADH are produced by oxidation of the 2 pyruvates? We're still with 2, right? And then our citric acid cycle. Each molecule of pyruvate that is oxidized enters the citric acid cycle and generates 3 NADHs, but we have 2 of those pyruvates per molecule of glucose. So the yield from the citric acid cycle is 6 more NADH. And then what else? Where else do electrons, where are electrons carried away in the citric acid cycle? How many of those? And what's the molecule? We have NADH, we also have what? FADH2, right? 1 FADH2 per turn of the cycle. We have 2 turns of the cycle per molecule of glucose. So we've got a total of 10 NADHs and 2 FADH2s. Those electrons transferred along the electron transport chain drive the proton pumping that results in synthesis of ATP. And we can correlate per molecule of NADH correlates or corresponds to the production of about 2.5 ATPs. The FADH corresponds to about 1.5 ATPs. And really what we're saying is FADH electrons, remember, they enter the electron transport chain a little bit farther downstream than the electrons that are passed from NADH. Therefore, FADH2 electrons drive fewer protons across the membrane. Therefore, those electrons correspond to a little bit less of that oxidative phosphorylation. So if we add up all of the ATPs that are generated from the oxidative phosphorylation, we have 25 from NADH, 3 from FADH2, add it all up, 32 ATPs per molecule of glucose. However, most cell types, remember, we have to get electrons from glycolysis, which occurs in the cytoplasm. Those electrons have to get into the mitochondria, and they have to be shuttled across the mitochondrial membrane. And so in most cells, NADH, the electrons from NADH are going to be transported across the mitochondria, and they're passed to FADH2. And therefore, those electrons correspond to 1.5 ATP each rather than 2.5 ATP each, which results in the total yield of 30 ATP. So that's why whenever we talk about cell respiration in any of our courses, we say 30 or 32, depending on the cell type. The difference is how much ATP does the NADH from glycolysis contribute to the total yield of ATP. All right. So we're adding up all our ATP here. We've got 4 plus 25 plus 3, 32 ATP. Now let's compare the yield of ATP from aerobic respiration and compare that to fermentation. Remember, fermentation occurs in the absence of oxygen, and so all of the ATP arises just from glycolysis. So how much ATP is generated by glycolysis? Per molecule of glucose, 2. We're back to 2 again. And how many NADHs from glycolysis? We're back to our 2 again. Thank you. But at the end of glycolysis, that pyruvate is not oxidized anymore, but the pyruvate has to be removed. And so it's reduced instead of oxidized. It's reduced to either lactate or it's reduced to, in 2 steps, to ethanol, right? So that the pyruvate is removed from the pathway, preventing glycolysis to reach equilibrium. As long as we have enough glucose coming into the cell and pyruvate is removed, glycolysis can keep moving forward. Right? It can be an overall spontaneous reaction. And glycolysis can only keep continuing as long as the NADH is oxidized. Right? It has to be, the cell has to be able to regenerate the NAD plus in order to allow glycolysis to keep moving forward. The NAD plus to become reduced as it oxidizes the glucose derivative, glyceraldehyde 3-phosphate. So total per round of glucose, 2 ATPs, but that's okay. That's better than not being able to make any ATP at all. So the cell, less efficient, but can generate ATP in the absence of oxygen, even in a prokaryotic cell with no structures for generating a electron transport chain, as well as pumping of protons. No oxidative phosphorylation, but as long as glycolysis keeps running, the cell can generate ATP for its needs. So let's remind ourselves of a couple of features of this process of respiration. In aerobic respiration, this is the reminder of what you should know. Overall cellular respiration, aerobic respiration, how would we divide it into 3 stages? What's going on in each of those stages, right? Glycolysis, citric acid cycle, oxidative phosphorylation. Where do all of those processes occur? What's the input? What's the output of each of those processes? What's the role of the electron transport chain? Where are the components of the electron transport chain located? How is a proton gradient generated? And what's the difference between fermentation and aerobic respiration? Think about answering those questions as if this was your test, right? It was an essay exam. Could you explain respiration in these terms to someone else? Could you write a 1-paragraph description of each of these questions? Think about that. If you can explain these things, then you know your material, right? And you're ready for our exam. So let's try to break it down to a couple of different types of questions. Thinking about and starting with really thinking about what's coming in, what's coming out. What is the overall yield of these different stages of respiration? For example, which of these steps or processes is going to produce carbon dioxide? Let's think about what is carbon dioxide, right? It's a waste product, but it is a fully oxidized carbon atom, right? That is an oxidized product of this respiration, this breakdown of that glucose that we started with. So which of these? Is there more than one?
Professor?
Mm-hmm.
My clicker, it says I have no response for question 2, but you just started it.
I maybe you need to I don't know what goes on in the clicker world, sorry. Maybe you need to restart it or something?
Yeah, I'm gonna try to refresh it. I just wanted to let you know.
Log out and log in is a recommendation?
Okay, yeah, give me a second.
Okay, if you have one problem with a clicker, remember your participation points come from as long as you have answered at least all but one question, right? Then you'll get your participation points. All right, let me close it up. I'll give you 5 more seconds. Okay. So of these choices, yes, only one of these steps is going to generate ATP, right? Breakdown of glycogen, well, glycogen is a polymer of glucose molecules, so it's a storage polysaccharide. Breaking down of glycogen is not going to yield any carbon dioxide. Glycolysis, that as a process, does not generate any carbon dioxide, it generates pyruvate. But when pyruvate, that 3-carbon pyruvate, is oxidized to yield the acetyl group that's part of acetyl-CoA, that's only 2 carbons, that fully oxidized carbon is released as carbon dioxide. And no carbon dioxide is released in oxidative phosphorylation. So what's the other step or process where carbon dioxide is produced? That's not listed here. Where's the last part? Right, citric acid cycle also generates carbon dioxide, right? The rest of the carbons are oxidized in the citric acid cycle. 2 carbon dioxides for each round, total of 4. There's all the carbons that started in, we don't have glucose here, started in glucose, the 6 carbons of glucose. Through this process, all 6 of them are released as fully oxidized carbon dioxide molecules. All right, so what about another feature of our cellular respiration? The oxidative phosphorylation portion of cell respiration. So what do we think? What happens in the final electron transfer event? Everybody have a chance to vote? Let me close it up. Oxygen is the final electron acceptor. The oxygen that was held in the cytochrome oxidase, waiting for the electrons at the end of the chain to be passed to it, that oxygen accepts those electrons, combines with some protons in the matrix, and the oxygen is reduced to H2O. Right? It accepts electrons, it is reduced. It's the final electron. It is the final electron acceptor. Right? The other electron acceptors in the chain are metal ions. They are iron atoms and sulfur atoms in different steps. You don't need to know where those are, but the electrons are actually moving through, being accepted and being donated by different metal atoms in the respiratory complexes. All right, so what I'd like to do, we still have a few more minutes here, is go through a couple more questions, pretty much one question from each of our other 3 chapters that will be covered on exam 2. So our next clicker question will be a question from chapter 9. See if you can answer these from without bothering to look through your notes. See what you can remember from your studying, your preparation from chapter 9. What about a hierarchical trait? Where does that arise from? How's everybody doing? Everyone have a chance to vote? All right, let me close it up. Right, so that mutation has to be able to be passed on to the next generation, right? And those germline cells, yes, they have to undergo mitosis first, and then those will be induced to undergo meiosis to generate the gametes that can carry that mutation to the next generation. Right? Mutation in the somatic cell is not going to be passed on to the next generation. A mutation during cell differentiation, if we're talking about in the body of the organism, the somatic tissues would remain in the organism that was mutated. But a mutation in a germline cell, either early on or later in meiosis, would be passed on to the next generation. All right, how about a question from our membrane chapters? From chapters 11 and 12, which of these statements is true? So what you want to do is try to find out or identify something that's wrong in 3 of the statements to identify which one is true. Is people still having problems with Reef out there? I see lots of questions going by.
Oh, it's just that I didn't see that you clicked the correct answer for the past question.
Okay, but I told you what the correct answer is. If I didn't click on it, I told you, right?
Yeah, yes. But it just didn't like show up on my screen as the correct answer.
Okay.
Yeah.
Remember, the grades in Reef are not the grades that go into web courses. So it doesn't really matter what your screen says, right? You're using your Reef to vote for me because I grade some of these and I don't grade others, right? So whatever Reef tells you as a grade is irrelevant, right? What matters is what I put into which of the questions I grade, because I only grade a couple of them. So you guys, I hope you guys are focusing on what is the right answer. Don't worry about the points at this point. I really want you to be focusing on the questions and why is A, B, C, or D the correct answer, okay? All right, everybody had a chance to vote? All right, let me close it up. So the one statement that is true is, let's see, is A. Only A is true. So let's find out what is wrong with some of the other statements. So A is true. A carrier protein can carry a solute across a membrane. It could be either direction, right? We said that they have this multiple conformation states, and we saw an example of a glucose transporter. If there's more glucose on one side of the membrane, it will bind, and when the carrier protein changes its shape, it will then be deposited to the other side. Right? And the net direction of transport will depend on the difference in concentration on both sides. So that is correct. What's wrong with B is pumps are transporters that can drive movement of solutes, right? Drive solutes from a region of low concentration to high concentration against the concentration gradient, but they require ATP in order to do that, right? Because the pump will actually phosphorylate itself and then remove that phosphate to change the conformation like that sodium potassium pump. So it doesn't synthesize ATP, it needs ATP. So that's incorrect. What's wrong with C? Let's see, facilitated diffusion is movement down its concentration gradient. that would be, that first half of the sentence would be correct. There are types of coupled transport, but that's a different situation, which is a type of active transport. Right? So we're asking about what is facilitated diffusion. Diffusion always is movement from high concentration to low concentration, right? Random movement of molecules to fill up the space that they are in. And then D is incorrect. Glucose transporter is not a channel protein, it is a carrier protein. Okay. All right, and one more question. We have a question from chapter 3. Here we're looking at the requirement for an activated intermediate, meaning a high energy intermediate, to drive this synthesis reaction, a condensation or dehydration synthesis. So we're asking the steps that are required in converting a glutamic acid to a glutamine. What's going to happen? We first have created the activated intermediate, and then the condensation step occurs. So what are we referring to here? Right, remember we described how a non-spontaneous reaction can be driven in the forward direction by coupling it to a spontaneous reaction that yields more energy than the condensation will require. Everybody had a chance to vote? 5 more seconds. All right, let me close it up. And the correct answer is B. What we don't see here is the activated intermediate, right? Glutamic acid to glutamine requires energy. What do we mean by that? Is that the glutamic acid has to be converted to an activated intermediate, a more unstable, a high energy molecule, and that utilizes the phosphate from ATP hydrolysis. So ATP hydrolysis will replace this hydroxyl with a phosphate that converts glutamic acid to a phosphorylated activated intermediate. And then the phosphate that was at this position can easily be replaced by this amine group from ammonia. Right? That condensation step is what we're asking about. Replacement of the phosphate that had been bound to this carbon, replaced in the second step with the amine from an ammonium. Right? Overall favored reaction as long as we couple the conversion of glutamic acid to glutamine, we couple it with ATP hydrolysis. So go back and refresh your memory about that process that we talked about in the coupling, how a spontaneous reaction can drive a non-spontaneous reaction. All right, so I hope that these kinds of questions will remind you of the kinds of processes and concepts that I want you to know, and I want you to show me your understanding in our exam. So think about explaining some of these processes to someone else. Think about summarizing each chapter by chapter. What in chapter 3, what are we talking about in energy? What are energy conversions? What's the difference between spontaneous and non-spontaneous? Why do cells need energy? Why do chemical reactions not come to equilibrium in a living cell? Right? Think about those kinds of things. Right? Not just memorizing individual PowerPoint slides, but what is it? What's the overall concept that we're talking about in each of these chapters? Okay. All right. Well, I hope you will be reviewing over the weekend, that you've been learning these all these chapters, one chapter at a time. Might want to consider taking a peek at your textbook to clarify if you have some, if there's some concepts that you're not clear about. And I will see you 9:30 Monday morning. I'll be in the chat if anyone has any questions. All right. Have a good weekend.
Dr. Bia, I have a question
Yes.
about this one. So you were saying that condensation, the step isn't favored, which is why it's coupled with the ATP hydrolysis. Is that correct?
Well, not the condensation step. The overall reaction is not. The overall reaction is favored because because of the intermediate that is formed, as long as the intermediate can be formed, which is coupled to hydrolysis of ATP.
Oh, okay. So if it's like an unfavorable reaction is coupled to a favorable reaction, it'll go in the forward direction?
Correct. Yes,
Okay.
you got it.
Okay, thank you. Have a good weekend.
Okay.